Welcome to Chicago, and thanks to Northwestern for hosting us, and for all of you making the long trek or short trek into the United States and or to Chicago. I'm Stephen Young. I have the honor of serving as President and CEO of SARC for the last 5 years, stepping into big shoes of an organization that's been founded since 2003. So good morning. We have a busy agenda today, so I'm going to just jump right in. A couple of late additions to the program. We have 3 new talks that are added in here. Jeff Kramer for the Conjugates Sarcoma Foundation is going to briefly talk about smashing sarcoma, which you'll learn more about. And also Dr. Sandra D'Angelo will be talking about the SFT registry and biobank program that they're working at the University of Miami. And finally, we're doing a spotlight of the National Leiomyosarcoma Foundation that you'll hear about. And part of SARC's goal is by adding more patient foundation voices into this meeting without overwhelming it or warping its primary mission. So if any of the foundations in the room would like to be spotlighted in future meetings, either at CTOS or ASCO, please reach out to me and I'll be happy to make that happen. First, I want to announce, we all were obviously devastated by Brian's loss, and I'm not going to regurgitate that. I'm going to look at what the positive outcomes of how much everybody loved and admired Brian. So we announced in December our new travel award called the Brian Van Tine Early Career Travel Award. And it's open to early career trainees and early career investigators at primarily academic sites. It's open to anybody in the world. It doesn't have to be United States. It includes $1,000 to help support, facilitate younger people getting to these kinds of meetings, the SARC meetings at ASCO and CTOS, but also to the parent or sister meetings, meaning the main part of ASCO. And in addition, and Dr. Fletcher could talk more about this maybe another time, a lot of SARC-associated meetings, such as the SAS, Strategic Advancement in Sarcoma Science, and critically the SASE breakout group aimed at the young folks. So there was a couple of layups, if you will, very logical places for these young folks to come in. So please let us know. And then I'm not sure how, is sarcoma P31 being talked about publicly, Jonathan? Okay, I want to make sure. It's up on the slide. Yeah, exactly, thank you. So in Heidelberg, Germany later this year, there's hopefully a very fascinating meeting, and I'm not qualified to talk about it yet. So moving on, if the first 4 awardees, if you're in the room, could you please stand up and everybody just say hello. Thank you for applying. Thank you. Finding photos of you, Dr. Sears, was not easy, so you're pretty invisible. I found one. If anybody would care to support this group, as I said, we had an anonymous donor who got the ball started, and several of you, Brian's peers, contributed to this as well. We want to keep this going on an ongoing basis. So there's a QR code there, and if it's too soon to get the QR code, just send an email or give me a call. First, I'm going to put the acknowledgments for the meeting up front because invariably we run out of time at the end, and this meeting could not have happened with a lot of key folks. One of which, of course, several of which are our industry sponsors, supporters. And I want to call out Adi Biosciences and Decipherer. I'm trying not to say Decipherer, Decipherer, to step up and support this meeting as our platinum sponsors. Our gold sponsors are Cogent Biosciences and Iovance Biotherapeutics. And our silver sponsors, for the sake of time, I won't read them, but you can see them on the screen. We're grateful to every one of these companies for stepping up and making this meeting happen. But critically, the one person that really has to be called out is Dr. Seth Pollack. And the difference of holding the meeting here versus the highest centric is not only is this much more cost effective, but it's obviously great to be in a medical center. So thank you, Seth, and to the entire Northwestern team. Seth is going to say a few words in a moment. Moving on, there are 2 other people I have to thank, and you don't have to give applause. Claudia Hill, our SARC's Chief Operating Officer, and Kim Ward, whose title doesn't describe what she does. She and Claudia basically handled 98% of the logistics of this meeting on our side of the fence. So I thank Claudia and Kim who are in the room. And I'm going to close here. I mentioned SARC was founded many of you know, back in 2003. And since then, SARC has raised and invested $100 million, a little bit north of $100 million in sarcoma research since founding. As you can see, we've sponsored and held the IND or collaborated on 32 investigator-initiated sponsored trials in the US and internationally. We have 24 that are completely completed. 6 are active and very much in the stage of either closing out enrollment and/or doing data analysis ready for publication. And 2 more trials we're launching later this year. And I'm going to highlight that for a moment. We're trying to, SARC is endeavoring to increase our throughput. On average, SARC has launched about 1 new trial a year since founding. We're trying to get into more of a run rate of 2 to 3 or maybe even 4 new trials a year. So if you have ideas for a concept or a study, please approach Dr. Scott Acuna, our CMO, or if it's a sciencey focused study, Dr. Fletcher, or our clinical trials development committee, which is chaired by Greg Cote along with Libby Davis. So a lot of avenues to get involved in SARC. And with that, I'm going to turn it over to Dr. Pollack to welcome all of us to his house.
Thanks, Steve. Yeah, just really quick, it's such a pleasure for us to host SARC. Again, last year was a fantastic session. This year, I'm really excited, and SARC has been such an important part of my career. I got a career development award from SARC when I was just starting out, and that really, I don't think I could have made it to where I am without that. So that was a really important thing for me, and I'm really excited to have everyone here at Northwestern, and you can see how amazing our facilities and hospitals and conference rooms are, and really just great to have everyone. Thank you.
I'd like to add my delighted welcome, Jonathan Fletcher, and Stephen, you laid out the groundwork beautifully, so I'm just going to show a few slides in way of thanks to some of our wonderful committees and to spread a little awareness of them. This is our committee that focuses on basic science, translational science, the Discovery Translational Committee. They do a lot for the conferences that SARC participates in, the pre-ASCO this and the pre-CTOS. But in addition, they have fostered a number of key conferences that Stephen has already mentioned. And part of that, we see growing opportunity to bring people together, which has always been the idea with SARC. And in the past 2 years, we have a meeting called SAS that's held at the NCI every year. This year not, because we're focused on this other meeting called Sarcoma P31 in Heidelberg, which is a lot of work. But next year, SAS will be back, Strategic Advances in Sarcoma Science. And the very first year, the early career folks said, we want our own meeting, our own morning. And they took the moniker SASSY, which made great sense. And we've followed that through, and in Heidelberg with Sarcoma P31, they'll have an early career morning. I think it's a fantastic way for folks who are about half with an MD in their title, half with a PhD without a clinical degree in their title, to get to know each other, to form collaborations. It's just been a remarkable success. They design the meetings, and they've come up with some remarkably clever approaches, so it's not sort of same old, same old. And then lastly, our executive committee for SARC, called the SARC Steering Committee. This is a group that takes on a lot of challenges and opportunities, but if we have a sciencey session like the one that's coming up that informs clinical approaches, they take that often from the Discovery Translational Committee, the first group, and add some sparkle to it and make sure that it's going to be relevant for a very diverse group of constituents that we have here. So thank you to all the committee members who support this organization and who make these vibrant get-togethers for all of us. And with that, I'd like to turn the reins over to Bree Wilkie, known to most of you, head of sarcoma medical oncology at University of Colorado, for our first session, which is on selective deliveries. Thanks, Bree.
All right. Good morning, everybody. And I'd like to thank Dr. Fletcher on all of the SARC committees for the invitation to MC this session this morning. I'm super excited about our speakers, and I think we're going to learn a lot. So I figured I would just very briefly set the stage with the obligatory AI image, which we now all have to start our talks with. I don't think I'll have to translate this one very much for all of you in the room. This is sort of the historical perspective of how we used to have to tackle sarcoma, which was with the caveman style hammer, really trying to use blunt force to go after all of our diagnoses. But the goal, of course, is to be able to develop precision therapy where we're using fancier tools, a more sophisticated approach to apply the right tool for the right job at the right time for the individual patient. And I would argue that our approach to split rather than lump has actually paid off with successes over the last years, at least since I've been in my attending career. We've seen novel agents be approved based on sarcoma types, based on specific molecular groups of patients. And a lot of this has led towards design of drugs that are thought about in light of resistance mechanisms and clinical trials that are really targeted towards a mechanism and the particular prevalence of that alteration within that group of patients. And really target identification. We've seen a lot of development of novel targets, which we'll hear about today. And while I am not going to comment at all on any outcomes, I'll just put this list of presentations that are on the agenda for this year's ASCO meeting. Again, all of which really are precision focused, looking at specific tumors, new drugs, and again, looking at potential resistance mechanisms. And so as we move forward today, I would encourage you to not just think about targets, but now we're talking about delivery. So as you know, sarcomas are stromal neoplasms, right? We're dealing with a lot of issues that involve drug delivery, hypoxia, bad blood flow, stromal barriers, metabolic neutralization, et cetera. And so now we're talking about homing mechanisms. How do we optimize the delivery of which payload, and what are our strategies to get it where it needs to go? And so we'll hear today about antibody-drug conjugates, we'll hear about radiopharmaceuticals, but as you're thinking about this, there are other strategies, including using one of the most effective payloads, which can be our immune system. And there's novel technologies coming down the pipeline that are designed to get that where it needs to go as well for sarcoma. So very exciting. And with that, I will bring up our first speaker. I don't know where his slides are, but I'm pleased to welcome Dr. Richard Gorelick from MD Anderson, who is going to talk to us about antibody-drug conjugates for osteosarcomas.
So thank you, Bree, for setting the stage. Thank you, Jonathan, for the invitation. Thanks, Seth, for this great venue. I like it a lot better than the Hyatt. This is much nicer. And obviously really appreciate the opportunity to sort of kick off the meeting and the discussion of science. I am sure you're going to hear a lot more about antibody-drug conjugates during this ASCO meeting, but let's start with a talk on antibody-drug conjugates for osteosarcoma. I have a couple of disclosures, so just the obligatory disclosure slide. I don't think any of this impacts on what I'm sharing. So antibody-drug conjugates, just by way of introduction, have three components. There's an antibody that directs it to a protein target. There's a linker that connects it to basically a payload, which is the drug that's delivered to the target cell. And is typically a chemotherapy, but it's typically much more potent than typical chemotherapy. So thinking that a topoisomerase inhibitor delivered by an ADC is the same as doxorubicin or topotecan is a simplification. These are very, very toxic drugs that would have to be selectively delivered. I think one of the things that is driving the development of antibody-drug conjugates is they're modular. Once you have a payload and linker combination, which is often companies' focus, any antibody can be conjugated to that molecule and then basically developed as a novel drug. So it allows a lot of quick drug development using different antibodies to target the same payload linker combination to different tumor types. This slide is becoming ancient because it dates back to 2021, and the slope of that exponential curve keeps going up. If we look at the number of antibody-drug conjugates that are approved and licensed for different indications, they keep exploding, and this is something that's likely to continue. So it was actually about 4 or 5 years ago that our laboratory published a paper where we took an approach of profiling osteosarcoma PDX models with mass spectrometry, so quantified the proteins on the surface of the cell, integrated that with RNA sequencing data on osteosarcoma tumor samples, integrated it with normal expression, and used that to create a list of targets. We validated those targets to sort of see what had products that were in clinical development, what had products that had antibodies, and then basically created a list of targets that we wanted to investigate further and validate that in samples they truly had expression. Typical way of doing this is sort of looking at TMAs, tissue microarrays. We had a series of tissue microarrays available for osteosarcoma. Our first three candidate targets were, CD276, which is also known as B7H3, MRC2, and MMP14. And all of these proteins have different names. But using this pipeline, we were able to validate that a number of these targets were actually expressed on PDXs, expressed on patient samples, validating that these seem to be bona fide targets. Ultimately, some of these targets have sort of moved forward to clinical trials, and this is the results of the Artemis 2 study that was conducted in Beijing, but basically tested a B7H3 exatecan ADC in the treatment of osteosarcoma, and really promising data was obtained, showing actually responses which are kind of atypical in osteo, but to a greater extent, prolonged stabilization of disease. Just like everybody else, nowadays, you just take AI approaches to things. And if you look at sort of the ADC trials that are out there, there are three different ADC constructs that have been tested in osteosarcoma phase 2 trials. So you just ask ChatGPT to compare those three studies. And what it kind of shows you is that, glembatumab vedotin versus an LRRC15 targeting construct versus the Artemis trial, there's a progression in sort of seeing increases in therapeutic activity. And actually, if you look at ChatGPT, it attributed the first two, and I don't know if this part of it's all that accurate, to the fact that the protein target expression correlated to outcome with the LRC15, but not the GPNMB. And then it attributed, obviously, the bigger increase to the differences in payload, which I actually agree with. So one thing before we go too much further in this talk is you also have to realize with good payload, you're going to do better, but the target still matters. So this is a recent study, led out of COG by Damon Reed in the audience as the first author, looking at a HER2 targeting ADC with an appropriate linker and payload, meaning the same as some of the B7H3 constructs. You didn't see a lot of activity. And, I think in the osteosarcoma field, we recognize that HER2 is probably not an ideal target. And basically, the point is the payload doesn't correct for that. So if we look at the clinical ADCs in development, specifically for osteosarcoma, this graph changes almost daily. And, folate receptor alpha is now the subject of a trial in Seattle using CAR T cells. So I would say in some respects, that box is now a green box all the way to the right. Everything is moving quickly, but you will keep hearing throughout this meeting and future meetings, either preclinical, early clinical, or later clinical data on a variety of ADCs relevant to sarcomas, and in this particular case, osteosarcoma. Our own area of interest is really, we believe osteosarcoma is committed to a transcriptional signal associated with bone development. So we think, as the defining feature of osteosarcoma is the expression of osteoid. We think that transcriptional signature, just like leukemias that are committed to particular biologies, actually defines the tumor, and some of those commitments are relevant as therapies. Our particular target that we've spent the most time working on is a particular target called cell adhesion molecule 1. I wanted this talk to be an hour-long talk, but Jonathan said I only had 15 minutes. So, it's always like this, what data are you actually going to show? This just shows you the RNA expression in the top right box and shows you its correlation to expression in normal tissues, and basically tries to get across the point that it's expressed in samples on the bottom and not in normal tissues and in PDX models. This past year, we were very fortunate to get a sarcoma spore. One of the projects of that spore focuses on ADC development, and one of our areas of interest is really cell adhesion molecule 1. I am obviously revealing a lot that's contained in that spore, but it saved me time in making slides and basically lets you know we're heading towards bispecific and trials and all of those type of things. One of the things that you have to think about is as you construct a particular ADC and sort of do this in the homegrown way, the payload matters and the linker matters. Things that are cleavable versus non-cleavable have different properties. We keep moving through different GMP grade combinations of payloads and targets to identify what's optimal. What this is meant to show you is, particularly with exatecan constructs, where 6 milligrams per kilo is probably a realistic dose, you see a lot of impressive activity. And where you see it tested at 3 per kilo, you were seeing responses, so we didn't feel a need to go up. And picking what probably is the most exciting to show is what we generated was resistant models. So what you had seen in the prior slide is that the tumors respond exceedingly well. So if you treat them with subtherapeutic doses to generate resistance, it allows you to see how resistance emerges. And really the biggest point of it is as resistance emerges to these particular ADCs, we don't see any loss of expression of the cell adhesion molecule 1. Our inference is they're becoming resistant through resistance to the payload, again suggesting that this is a committed signature that can't be downregulated. As we move to trials, one of the things that's really promising is following these in blood samples. We have a particular interest in collaboration with Sam and Ash, also at MD Anderson, looking at exosomes, using exosomes as a marker of what happens to expression of protein targets. We intend to sort of use circulating tumor DNA to establish the level of tumors and an integration of that with the protein expression on the vesicles to track this in clinical trials. And really, I figured I would come to the end by just providing a couple of lessons. And one lesson that we learned is really the antibody matters a lot. What you're seeing on the left side of this panel is how people who develop antibodies think about different antibody classes. And what defines them is what epitope they bind to, affinity, and internalization. What you're seeing is the binning of various different B7H3 antibodies, some of them the commercial ones, some of them from other sources. And basically, antibodies, they're sort of an idiosyncratic of sometimes when they bind, they internalize, and sometimes they don't. If they internalize, they're relevant as an ADC target. If they don't, it's more, in the immunotherapy domain. The binding affinity influences the binding to normal tissues and sort of has a little bit of an integration with toxicity. And basically where it binds is important for resistance because you can cleave that or modify it. The biggest problem for the field overall is the number of payloads. There's not a lot of payloads out there. So if you think about them, most of them are microtubule inhibitors and more of them are sort of topoisomerase drugs now. We clearly need more. I mentioned tesserin, which isn't really a drug, as one of the compounds that you test preclinically. The problem is the alkylating agents are way too toxic, and they've been used for some hematopoietic malignancies, but for solid tumors, they're probably going to be too toxic. So clearly we need more payloads, we need more approaches, accepting that things like T-cell approaches and radionuclide approaches also are relevant. So again, I always talk about osteosarcoma, but I know this room, not everybody focuses on it. What I would suggest as a paradigm is you think about the payload first. So if in your sarcoma it doesn't respond to anything, then you have a problem. But if that sarcoma responds to something, whether it's a radionuclide or a drug, then you have, or an immunotherapy, you have an opportunity to think about whether there's a therapeutic that could target that surface protein. We actually think the model we provided, which is screening PDXs first, integrating with RNA-seq data, and then sort of selecting targets, actually is the way to go. And so, if I had one message for the sarcoma community overall, it's we need more proteomic profiling in sarcomas. It's been done in some, but not all, and should be done in more. So wrapping up, I think targeting surface proteins with antibody-drug conjugates is a promising path forward. Need to think about ADCs in 3 parts. I do think we're going to hear more and more about clinical trials of ADCs in osteosarcoma. And we think targeting bone transcriptional signatures makes a lot of sense. And we think they have relevance to other sarcomas. And there's a wide open field for proteomics and surface protein targeting. And before I wrap up, of course, I don't do anything. It's the people in my lab who do all the work, collaborators who come up with all the ideas, and I just get to present. And thank you all for listening. So I think you're doing questions at the end, right?
Yes, we'll just ask everyone to hold your questions for the end. Dr. Fletcher has arranged for a substantial discussion time in response to your feedback from prior years, so thank you. Our next speaker is going to be Dr. Zachary Morris from the University of Wisconsin, who's going to be talking with us about integrating radiopharmaceuticals in the combined modality treatment of patients with sarcoma. Dr. Morris.
Good morning. Pleasure to be here today. I really appreciate the invitation as a physician scientist who primarily sees patients with sarcomas. I have utmost regard for what this group has done and would love to talk with some of you about how I might get more involved after this. But appreciate the invitation, opportunity to speak. So building on the last talk, I'll be talking about radiopharmaceuticals in many similar ways. We are talking about delivering a payload of an isotope here. So take everything that was just stated about the importance of the target and the different approaches to target the surfaceome. But in this case, our linker leads to typically a chelator, which is binding an isotope. Some differences and potential opportunities that this field presents. You can have isotopes that emit imageable forms of radiation as well as therapeutic. And so with radiopharmaceuticals, we can do imaging to develop quantitative assessments of how well a drug is taken up in different tumor sites across the patient. And we can change the isotope. And as we'll hear from our next speaker, there's different isotopes such as alpha emitters that may change the range of an emission and also the potency. So with that, by way of a broad overview of radiopharmaceuticals, I'll jump right in. I do have a number of disclosures. One that is relevant, I'm on the advisory board for Arceus Technologies, and I'll talk about a drug that they are bringing to first in human. I'll be using it primarily just to illustrate a broader point, though. One thing that I wanted to start with is actually a clinical trial that we completed not long ago, led by some colleagues of mine, Grace Blitzer and Brad Uckelman as a resident who's finishing up and going to be at Michigan. And this was a study where we did hypofractionated radiation, or SBRT, to unresectable sarcomas, or in some cases, oligometastatic disease. And we treated fairly large tumors here. These were some of the true bad actors in terms of local and oligometastatic disease. With, you can see our target volume ranging up to 800 cc's. And this was simply a way to deliver external beam and achieve dose escalation. But what I find interesting is that our local control here, going out beyond 60 months, is upwards of 90%. And historically, comparing that back to data where we looked at delivering radiation to achieve local control for soft tissue sarcomas, that's quite an improvement over fractionated external beam radiation, where we were typically on the order of 50 to 60%. And the message I think that brings that is relevant to this talk is that soft tissue sarcomas can be locally controlled and potentially systemically controlled for a period of time if we can give enough radiation to achieve that, and dose escalation matters in this. And one way that we could achieve that is through the use of radiopharmaceuticals. And as was alluded to, there is the potential to develop novel radiopharmaceuticals for targets that are specific to each and every one of the different subtypes of sarcomas. And I think that will be extremely valuable, and those could be tremendous monotherapies. But it will take a long time to do that. And there's also the potential to target sarcomas and other cancers with mechanisms that may not be quite as tumor specific and may therefore sacrifice a degree of selectivity or ability to give dose escalation, but would have broader applicability in combinations with other therapies that are also not specific to one type of sarcoma, such as external beam radiation. And this drug is one example of that. Another drug that I'll talk about that has clinical interest in soft tissue sarcomas are the FAP-directed radiopharmaceuticals. These are drugs that target components of a tumor microenvironment. In this case, NM600 targets the alpha-phosphocholine enrichment of certain lipid types in the membranes of tumor cells compared to normal tissues. FAP targets fibroblasts within stromal dense tumors, such as many soft tissue sarcomas. And so this agent was developed by my colleagues, Jamie Weichert and Rainier Hernandez at the University of Wisconsin, and targets cholesterol-rich domains of membranes, which we do see uptake in human soft tissue sarcomas. And to illustrate what I think is an opportunity with these types of agents, which may not be specific to one type of sarcoma, but which can deliver radiation to a tumor microenvironment selectively. We ran a clinical trial. This is an example from head and neck cancer under our head and neck spore grant at the University of Wisconsin, where in recurrent head and neck cancer, these patients have often received prior radiation, and if they develop a recurrence, it's a very challenging circumstance where further radiation is often indicated, even to achieve palliative outcomes, but yet you're re-irradiating an area that's very sensitive to further treatment. And the opportunity that I think this illustrates is that many times in soft tissue sarcoma or bone sarcoma, we are looking to give radiation, and if we could give enough, we could probably achieve disease control, but we're limited by our organs at risk. And for external beam, those are almost exclusively the nearby tissues. However, adding a radiopharmaceutical from the tumor standpoint is simply adding additional radiation that allows effective dose escalation. But from the normal tissue standpoint, the off-target effects of most radiopharmaceuticals are the kidney, the liver, potentially the bone marrow and blood cells, which are often not a concern in terms of organs at risk from external beam when we're treating these patients. So it's an opportunity to get additive effects on tumor without additive effects on normal tissue. And so a way that we could do this in this study, we used patient-selective imaging to quantify uptake, perform dosimetry, and determine how much dose we could safely give of a radiopharmaceutical to a tumor in a patient where we were looking to give palliative radiation. And then we simply subtracted that amount from what we were giving with external beam and gave that with a radiopharmaceutical in a combination approach. That's a way to maximize the safety of a given treatment. Alternative approach would be to calculate how much we can give and give that on top of what we could give or intended to give with external beam. But I highlight this because I think it's an opportunity, given that radiation dose escalation increases tumor control in sarcomas, to deliver more dose than what we're currently doing in these patients with external beam alone. Another opportunity for these types of radiopharmaceuticals that target what are often immunogenically cold tumors in the setting of sarcoma is to use radiation in combination with immunotherapeutic approaches. Radiation kills predominantly through the induction of DNA damage, but for decades now, it has been recognized that a component of the killing of tumors following radiation therapy is mediated by immune response. And this is different than to say we're looking to target, for example, an abscopal effect. Rather, the target that we're treating with radiation responds better to that radiation in the setting of an intact immune system compared to if there is not an intact immune system. The immune response is an inherent part of the response to radiation therapy. However, in many ways, we're just beginning to understand how to optimize that immune response. Generally, what we have focused on is optimizing the direct DNA damage coming from radiation. And as we've begun to look at this in the lab, there's many different immunologic effects of radiation, and they're dose dependent. And it's not the case that highest doses of radiation necessarily optimize a given immune response. In fact, that's not the case for a number of parameters that you would potentially want to induce if you were looking to optimize immune response following radiation therapy. Low doses enhance certain upregulation of endothelial adhesion markers. Moderate doses enhance optimally the activation of type 1 interferon responses, and high doses optimize not only cell killing, but the release of damage-associated molecular patterns and the increase in MHC class 1. this highlights a potential opportunity for radiopharmaceuticals that was otherwise often considered a weakness. Compared to external beam, where we can uniformly deliver radiation spatially targeted across the tumor microenvironment, one potential limitation of radiopharmaceuticals from a strict radiobiological standpoint is that they're subject to the delivery through perfusion in blood and through the targeting of a certain molecule that might be heterogeneously expressed in the tumor microenvironment. And that can lead to regions that get effective doses of radiation and other regions in the same tumor that get ineffective or lower doses of radiation. And that could be modified by the range of the isotope. If you have a very short range, that's going to maximize the heterogeneity of dose in that tumor microenvironment. And if you have a longer range, it's going to kind of smear out the difference there. This has long been thought to be a weakness and a potential limitation of the field of radiopharmaceuticals, is that if you can't give radiation to a portion of the tumor, how can you expect to achieve response? But a really talented graduate student in our lab has been looking at this and has been challenging that paradigm. To look at this in a controlled way, we generated tumors that express FAP, which I mentioned is clinically relevant in the setting of sarcomas. And we generated tumors that either uniformly express FAP or those that have a very heterogeneous distribution of FAP that are composed of cells that either are positive or negative. And I would say from a classic radiobiological standpoint, I would have expected that a uniform distribution would respond better to a FAP-directed agent. But in fact, that's not what we saw. What we observed was that we achieved better local tumor control and better overall survival in these tumor models when we had a tumor that was composed of a mixed FAP-positive and FAP-negative population of tumor cells compared to those that had uniform distribution. And furthermore, what we observed was that response in the mixed tumor model was entirely dependent on the presence of CD8 T cells. And so it speaks to the potential that the response to radiation in certain settings may be dominated by that immune component as opposed to the DNA damage-based component that has been largely focused on in conventional radiobiological paradigms. When we zoom in and look at the tumor microenvironment following a heterogeneous distribution of radiation, what we see is an imprinting of the dose on the response as a molecular level in the tumor. You see the induction of DNA damage where you'd expect it. And so the IQID image here is a literal image of the radiation being emitted from this radioactive tumor. And then you look at areas of high activity versus low radioactivity, and you can see that in high areas, as you would expect, you induce more DNA damage. But you also can then see the induction of certain immune responses. And what was very interesting is that because, as I started out saying, dose response to radiation leads to different dose-dependent effects, with a homogeneous tumor, you can activate certain mechanisms with certain mean doses. You can activate a high-dose mechanism with a high mean dose. You can activate a moderate dose mechanism with a moderate dose, and so on. But with a heterogeneous distribution, you can activate all of those mechanisms in one single tumor microenvironment. And we believe that may be critical to the advantages that we see with heterogeneous distributions compared to homogeneous. In addition, heterogeneous distributions have cold regions of the tumor, which I would have thought might have been a weakness. However, from an immune standpoint, those create sanctuaries for immune cells to preserve their functionality. And what we see when we look at dendritic cells is that we do not deplete dendritic cells in a heterogeneous dose distribution in the tumor microenvironment. We preserve them and their ability to migrate to tumor-draining lymph nodes, whereas in a homogeneous setting, we impair that migration because of functional damage of radiation on those tumor-resident dendritic cells. And consequently, at a later time point than the following week, we see an increase in tumor infiltration by T cells, and we see clonal expansion of T cells consistent with antigen cross-presentation occurring in those tumor-draining lymph nodes following heterogeneous radiation, but not homogeneous radiation. We've tested the implications of this clinically. Pluvicto is a drug that is FDA approved for the treatment of men with castrate-resistant prostate cancer. And we took patients who we had treated at the University of Wisconsin for whom we had full dosimetry, and we broke them into two broad categories. One, where at any given tumor in their imaging, they had a tumor that showed a complete range from it that included low-dose regions and high-dose regions in one tumor microenvironment. And then alternatively, there were those with more homogeneous, where they either had low and moderate but no high dose in a tumor, or high and moderate, but no low dose preservation in tumors. And then we looked at the progression-free survival as well as the PSA response in these patients. And in support of our hypothesis that an immune-mediated component of a response may be at play in the basic response to radiopharmaceuticals alone, we had a better response in this heterogeneous distribution in contrast to the homogeneous distributions of radiation. And so our hypothesis is that in the setting of radiopharmaceuticals, there may be ways to maximize the immune-mediated destruction of a tumor by making sure that we deliver a dose that does not induce lymphopenia and that preserves low-dose regions in a tumor while also achieving high dose. And what I think this presents that is interesting is the opportunity to consider or reconsider what might be optimal or effective targets within a tumor microenvironment for the treatment of diseases where there's a high degree of heterogeneity. We might have in the past thrown out certain drugs because we didn't think we could cover the full tumor with radiation, and that may warrant revisiting. Here's an example of a preclinical KP sarcoma that we got from David Kirsch's group, where we see this tested and applied in the setting of soft tissue sarcoma models, where the optimal dose in this setting was not the maximum tolerated dose, but rather a dose that abided by those principles I just mentioned. And so this is, I think, a potentially clinically testable hypothesis about optimal dosing of radiopharmaceuticals. And taking that further, I would posit that the best way to deliver radiopharmaceuticals in combination with immunotherapy would be the way that maximized the immune response to that radiopharmaceutical, which again is likely not to be maximum tolerated doses, which is often what has been done in the selection of dose in this field. And just as a proof of concept for that, we've generated preclinical data looking at combinations with checkpoint blockade that I show here, where maximum dose was less effective than a slightly reduced dose. And similarly, in combination with CAR T cells, in this case, a GD2 CAR T cell. And I think there's enormous potential in the field of treatment of patients with soft tissue or bone sarcomas to further the roles of checkpoint blockade and CAR T cells. And it may be that combining those with a moderate or heterogeneous dose of radiation from radiopharmaceutical could help as a combined modality approach. So I'll close there and just offer concluding points that I think there's enormous opportunities for immediately looking at ways to add radiation in addition to radiation and get additive effects on a tumor without additive effects on normal tissues. And there's also a tremendous opportunity to use radiopharmaceuticals in combination with immunotherapeutics to potentially achieve synergistic outcomes. But the way that we deliver that radiopharmaceutical and the selection of agents may differ from what would otherwise have been predicted if we were simply trying to kill every tumor in that tumor microenvironment with radiation dose. So thank you again for the invitation. I want to thank all folks in my lab and collaborators again who made this work possible. Thanks.
So listen, great talks. Nikon, great to see you. Oh my goodness, it's been so long. I have a question for you though. GD2, GD3 osteosarcoma, great concept. A lot of heterogeneity. I think you showed that in a paper, the different heterogeneity even in tissues. So how do you address the heterogeneity of sarcoma with such types of different expression? And for GD2, should we be thinking about a GD2, GD3 SADA rather than just a GD2 SADA? Just want your thoughts on that.
I'll let him answer your question about heterogeneity and vaccination effect. But one of the things why we decide to use recombinant antibody is because these things are actually very small. So these, what we call the tandem single chain, can actually penetrate tumor extremely well. Why I'm saying that? Well, when we take a tumor that is 1,000 millimeter cube, these are big tumors in the mice. These tumors goes away with alpha. So what does it say? It said that every tumor, essentially, is probably getting something, and that the alpha, which works only nanometers, there are no cross-firing, is actually killing the tumor. So this is not a direct answer to your question, but when we do autoradiography, which is when you do this, there's an even distribution of this isotope. Now, again, depends on the tumor you're using. Maybe the tumor that we pick, the PDXs we have, are just very homogeneous, so I have to qualify that also. But I do think that your question is very important. is heterogeneity definitely important, but is that the only reason we should consider? But maybe the vaccination effect that you just mentioned is going to be so important.
Yeah, I'll just add, as I mentioned, I think there's some potential that other effects beyond direct, such as immune-mediated response, could help overcome challenges of heterogeneity with these. I'll also say that the heterogeneity that is related to perfusion differences, tumor uptake that's not target-specific, can sometimes be addressed through giving more than one cycle, and you change the tumor microenvironment with the first cycle and potentially open up opportunities. And with short-range agents, you may put more selective pressure on the loss of a target, but with longer-range isotopes, you may not, because you also can address heterogeneity through radiopharmaceuticals, in part because much of the damage to any given cell comes from its neighbors that have taken up the drug, not from the cell itself. And so there is some mechanism for overcoming that challenge just from a radiation killing perspective alone.
So is there a TME effect with your GD2 SADA? Have you looked at that? Is there a change in tumor microenvironment beyond a direct toxic effect?
We don't know that. We don't have any study on that, because we're looking at immune deficiency mice. So all we're looking at is delivery.
Okay.
We can't really ask that question that Zach is asking about the vaccination effect. Okay. But it is a possibility. You can do cocktailing. You can combine different isotopes at the same time. So you can do lutetium, actinium together, so that you can achieve the cross-firing effect for heterogeneous tumors, but also looking at alpha being the more potent.
Thank you very much.
Yeah, one more comment. So on ADCs, they largely come with linkers that are cleavable and non-cleavable. The concept behind the cleavable is you get a bystander effect because there's local diffusion of drug. So that's another way of capturing treatment in the face of heterogeneity.
Okay, there's a giant light behind your head, so I have no idea who you are, but please go ahead.
No worries. This is Andy Brohl-Moffitt.
Thank you.
Thank you for the nice talks, first of all. My question's for Dr. Gorlick. You mentioned the need for additional proteomics for target identification for sarcomas more broadly. And so I was curious of your thoughts to the concordance versus discordance of RNA-seq data versus high-throughput proteomics data like mass spec or whatever. And as a follow-up question to that, if you were advising one of us that has a sarcoma of interest and that has RNA-seq data that shows overexpression of these 10 cell surface protein targets, would it be reasonable to start with those 10 and validate those? Or would you advise me to not even look at the RNA-seq data because it's just going to get me down a rabbit hole and just do mass spec as a starting point?
You're correct. There is some discordance between protein and RNA data. It's not as big as you sometimes believe it to be. I think our approach, what our focus was we didn't need to eradicate every false positive, and we didn't eradicate every false negative. We just needed a couple of bona fide targets. So as long as you have a couple where there's concordance, it's validated, you're good. I had a slide actually that I chose not to share, not because of time, but because I thought it was a little bit too bombastic. And when you look across sarcomas, there's varying focuses. So, if you think about Ewing sarcoma, there's a lot of translocation targeted therapy, translocation-oriented research. Maybe surface protein hasn't been looked at to the same extent. If you look at some like synovial, it's sort of both, but it's kind of all over the map what gets researched in each malignancy. And I kind of think each sarcoma needs a little bit of crosstalk that maybe is benefited by meetings like this and CTOS, but we need a little bit more crosstalk. I think I think your surface protein data off of PDXs is your best starting point. So we started with protein first and then used RNA to narrow it down. I think the problem with starting with RNA is you get a lot of false positives and a lot of false negatives. So I would start with surface proteomics on cell lines or PDXs and then use the RNA-seq to whittle it down.
And you think that's a superior approach than using patient samples, if patient samples are available?
So it is hard to do comprehensive proteomics on patient samples. You just don't have enough volume of material typically. So again, that's why we emphasize PDX models where it seems to be relatively consistent with samples, and then use patient samples to validate, of course.
Thank you.
If I might to add something, I think it's a very important point because B7-H3, the message is ubiquitous. Every tissue has it. So if I use mRNA, I would have rejected that target right off the bat. So proteomics, on the other hand, is extremely restricted. And it turns out there was a really tight control. What we showed years ago was the microRNA 29 A, B, and C actually control the message that this B7-H3 has in the cell. And certain tumors have more of it or less of it. And one thing that was striking is when we look at brain mets of many of these tumors is that particularly was a big differential between brain and a tumor, so that many of these tumors actually has high levels of B7-H3 expression at the protein level when they metastasize to the brain.
Dr. Elias.
Yeah, I dimmed my halo so you could recognize me. The issue of payloads, of course, is a major problem. But I would also like to emphasize that we've learned something from chemotherapy, namely that combination therapy is a lot less likely to develop resistance and more likely to be curative as long as one uses the mantra of no overlapping toxicities, et cetera, and that might be eradicated. But the point being that all the pharmaceutical companies currently are using one nail in all of their targets. The targets don't disappear, but there's increasing data to say that in the clinic, sequential ADCs, the resistance is at the payload level.
Yeah, so trying to take that on, I think everybody knows the next frontier for ADCs is really combining them. And I think you're starting to see evidence that companies are beginning to think about sort of sequential ADC trials or combination ADC trials in different forms. I think what's getting really challenging is sort of getting at some of the preclinical work we're doing. It's trying to understand, do you want to use multiple antibodies with the same payload? Do you want to use multiple payloads with, same antibody, different antibodies, different payloads, bispecifics? And I think the combinatorial part of it's going to require some preclinical work just to sort of know how to combine it best. I think that coupled to a little bit of toxicity fear has sort of slowed the field a little bit, but you're absolutely right. One ADC is never going to cure a patient. You're going to have to figure out ways of combining multiple ADCs, perhaps with systemic chemotherapy. That's actually the first trials are actually, ADCs plus drug. They're a little bit simpler, a little less elegant, but you're absolutely correct.
So I think that's what's so exciting about your technology that you're showing is that you can put two different particles in there, and if you can get enough of it in the neighborhood, can we bomb with maybe not the perfect antibody or the perfect homing device, but just the right payload?
I just might also want to add that what we saw very preliminary is when we look at recurrent tumors after these treatment, we saw a change in the lineage. So this is whole idea of lineage plasticity, which is playing out in here. And they have different actually expression of certain antigens. And this, I think, is very important, is to look at resistant tumors or recurrent tumors to identify what are these targets or targetable proteins that they now adopt. Prostate cancer, for example, you can see a change in the lineage. Colon cancer, you can see a change in the lineage. I think those are very important questions. They require a lot more work. Again, it's not as easy as you would like, but it's something very important.
I think one area to maybe bring the two drugs together here, or opportunities, if you don't have a bispecific, you may be able to achieve bispecific-like selectivity and widen that therapeutic window with sequential use of individual agents, particularly if you target induced targets. And so one of the targets that we see go up proportional to dose of radiation is B7H3. And so, for example, if you were to combine a GD2 targeted agent, but that induction takes place transcriptionally, so it takes a few days. And so you wouldn't want necessarily to have a bispecific GD2 B7H3 at the same time, but a follow-on with a different drug. But that does open the opportunity to potentially induce targets that might not otherwise be selective, but to do it spatially restricted so that you can enhance your drug.
Okay. This is Kamlesh Sankhala, Next Gen Oncology, Los Angeles. My area of focus is drug development in sarcomas. And my question about the ADC in osteosarcoma is, ADC is a big molecule, and you're dealing with a dense microenvironment with extracellular matrix in the bones, and that's true for all bone tumors. How do you address that pharmacokinetic issues there?
Great question. So you're asking the question of delivery of ADCs as a large molecule into the microenvironment. And I think there's a lot of work that needs to be done in osteosarcoma to understand sort of the landscape. We are a little behind in sort of the spatial transcriptomics part of the world. I think we're starting to see some data emerge. I think most of what's out there is really about the immune landscape and the T cell exclusion, much less about vascular permeability. I know the orthopedic surgeons are always interested in sort of the pressure issue in the context of tumors with their lacking lymphatics and bone. I see Dr. Weiss right behind you. I'm going to defer to folks like that on those sort of issues. I do think what ameliorates it partly is most of the time in the relapse setting, we're dealing with pulmonary metastasis, and the dynamics there aren't the same. That said, do we actually know what the dynamics are? Not really. Does it look like we're getting drug delivery just based on effectiveness? Yes, but there's a lot more to be known.
Yeah, because these looks wonderful in the lab setting, but when it comes to the human trial, we'll face those issues. Thank you.
Kurt Weiss, Pittsburgh. That was actually what Dr. Chung just said about plasticity and what you shared, Richard, were perfect segues to my question. Thank you, first of all, for your talks. I learned so much. But I try to be a metastasis guy. And I appreciate that you're just trying to figure out how these tools work with the linkers and the payloads and all the things. Have you done any work specifically looking at metastatic disease, and is that on your menu as you proceed? Because I feel like we got a good answer for what to do about the tumor in the arm or the leg, and that's my knife. What we don't have a good answer for is metastatic disease. So is that where you're headed?
That's a complicated question. So your question is really about metastasis and concordance versus discordance of response and work we've done. So in that area, we actually have tested the B7H3 ADCs and sort of metastasis PDX models, just as sort of a validation that we're going to see the same kind of efficacy there. That was actually presented at AACR just, recently, like a month or so ago, and we didn't see much discordance in response. A lot of people will believe that metastases are biologically different from the primary. I'm not as sure that it's a stochastic type thing versus there's a true difference in the biology of the tumor itself. The microenvironment's different. And again, it comes back to the same answer. I don't think we understand the landscape of the normal microenvironment in the context of tumor in the bone versus lung. And I'm not sure whether it's that or differences in the tumor. Most of what we're seeing from profiling work is it's not the tumor. It's the microenvironment that's different. But I agree. The lung is what matters as a sort of a tumor site.
Thank you.
Maybe you guys have a better answer.
The only thing I might add is just from the perspective of someone who completely agrees with you about the challenge of metastatic disease, but local therapies, I think do offer some potential. I think one thing that would be interesting is to begin seeing the use of more diagnostic imaging or predictive imaging in the field of ADCs and get a three-dimensional spatial understanding of where we can get predictive tumor-specific response, because you do have a number of partners with the knife, the beam, or ablative approaches that could take care of a select number of metastatic sites and salvage an otherwise effective treatment.
Amen. Pictures or data, we should use them more. Thank you.
Dr. Fletcher.
Yeah, I might have two questions, but it depends whether anybody else stands up, which to start with. I'm going to start with the easier one, I think, which is using mice, okay? Yes. Not everybody in the room might, oh, super. Not everybody in the room might know that the NIH has vigorously recently encouraged lab researchers to dial down substantially the use of mice. And listening to these three talks, it used to be DNA damaging agents, combinations. We wanted to see the tumor regress and the mouse was frisky. And Dr. Chung is raising the bar substantially, saying, I want durable cure for 6 months, minimal toxicity, right? And so, my question is like partly facetious, but not really. It's if as the NIH, I've been going back and forth with them personally, trying to gain some traction and saying, but, so if it's true that with 3D models, other in vitro approaches, that one can approximate some of this, then do you see your dependence on the mouse models increasing because there's so much opportunity? Or do you actually see that in absolute numbers, you might be able to use less in vivo systems to chart the way forward?
It's a very important question. It's also a tough question. If you skip the mouse, skip the monkey, you're going to test it in patients. So that's the first thing. Who's going to suffer? Patients. Because modalities are not necessarily predictable, I think, and particularly in pharmacokinetics. It's very hard to predict. The microenvironment is very hard to predict. And I don't think we know enough, maybe, might put it the other way, to recreate that in a in vitro model. I think it's take a little time, not that it's not doable. I think we are not quite there yet. So you're going to make some assumptions, and the risk is going to be passing on to the patient. I think it's a very hard thing to say. Maybe it's true for some systems, maybe it's not so true for other systems. So imagine this is a pediatric patient that we deal with. I think it might be one, I don't know, you want to set the bar a little bit different. These are kids that, sometimes I don't know. I think that just say, it depends on the context, I would say. I would certainly like to get rid of the monkey studies because that's, a lot of times, it's not even predictable. Second, it's costly. And it just add another year or two to the whole drug development pathway. I agree with that. If we can avoid that or skip that.
What about dogs? So if you, do your antibodies cross-react with canine? Because at least with osteo, many of us in the room have been excited about the potential of actually treating our little pets and everything that have osteosarcoma with the same sort of agents that might then give you a little more real-world.
No, I think I agree, right. I think you can always pick another animal model, but sometimes it's harder to get an ILB approved in animal studies than in a human study. So you have to keep that in mind also. It's not just you want to do it. Mm-hmm. You have to have that hospital next to you or the collaborative centers. So I think it's a good idea to use another animal model, whether it's this dog or pig or whatever. But as someone who, I've been doing this for 4 decades, trying to get antibodies from the test tube all the way to the clinic. It's just a lot of work to get that to, big companies, yes, they have billions to spend. Academic, I think it's a little tough. As I said, the monkey part was already difficult. If you contract it out, it's going to cost you a couple $1 million just to get that primate study done. And at Memorial, we had monkeys, but it's very difficult to do anything. As I said, the monkeys need to be, number one, housed in a special environment. They have to have TVs every day. They have 3 meals a day. There are many things that you have to do to keep the colony up to spec, right? So it's very hard to do that kind of study in academic setting. There's institutions who have those, I don't know about MD Anderson facilities, but it's hard. It's very hard. And you need numbers. I mean, the NIH is not going to take 2 months. I mean, we had a study where 4 monkeys, we said no good. You need to do the whole thing, right? So it's something very difficult. It delay the drug development. It all adds another cost to it, which is already expensive. So anyway, I don't know whether I can answer your question or not.
Hello, my name is Langdon Miller. I'm from a small company, Solve Therapeutics, and we're developing a theragnostic approach to ADC development, just what you mentioned, where we're using an antibody with a zirconium-89 PET positron emitter to try to do PET imaging of patients to identify whether the target is present before we administer the exatecan-containing ADC. So the hope would be that, in fact, we can select patients that way. The difficulty, of course, comes that the patients may be resistant to the payload, or the tumors in the patients resistant to the payload. And Dr. Gorelick, I guess I was wondering, based on the data you showed about resistance, do you find that there's any particular signature that would suggest a way to identify patients who are likely to have exatecan-resistant tumors a priori, and so one could select away from those patients in terms of administering them an exatecan-containing ADC?
Yeah, let me start by saying I think it's phenomenal using the approach that you radio label an antibody target to know whether the antibody gets to where you're going and basically starts assessing, is this a relevant target in vivo? So I think all of those efforts are phenomenal. I think I hand wave that the resistance is to the payload, partly because we've done molecular profiling of those tumor specimens to try to understand what the basis of the resistance is. So far, it's kind of all over the map and can be everything from sort of classic, export proteins through, repair pathways. Particularly for osteosarcoma, which is very heterogeneous and has a lot of genomic complexity. There's so far not really a consistent single mechanism of resistance that defines it. Answering, there's not a good target. Obviously, having the protein and having the protein there and your antibody able to get to it, that's a good sign. Right. But it may not be perfectly predictive because of the other elements.
Yeah. Thank you. And Dr. Chung, I'm curious, we're at Solve Therapeutics very interested in astatine-211 as an alpha emitter and its potential application therapeutically in antibody radionuclide conjugates. Have you done any work with astatine-211, and would you see it as being feasible with your approach?
Yeah, definitely. Any alpha. The only limitation is we don't have a production and MSK to ship the astatine. It's going to take, a day maybe. Yeah. And you're going to lose a lot of that within that time period. I think the reasons we are picking actinium-225 is because in the real world, you're going to get delays in the shipment. You're going to get all kinds of, and by the time you have a delay, your isotope is gone, so to speak. Sure. So even lead-212, the half-life is actually relatively short, even though it's a great isotope. Yeah. We do have a production on site, so that's a different story. But for most places, when you don't have a production of the lead on site, you have to ship it from outside. Yeah. And again, as I said, I don't know how good the mail system is, but you always get weather issues and they get delayed, and that's it. You're done. So I think that's some of the consideration that we have. It's a supply chain. Yeah. How are we going to get this supply chain resolved? But again, astatine, yes. Why not? But if you can get it.
And the issue being that astatine has a physical half-life of about 7 hours. So it does make it difficult, to actually get it there. And that has been the fundamental problem with developing this concept, though it does have many potential advantages over actinium. So we're hopeful to get there someday, and I look forward to maybe applying this technology with what we're doing. Thank you.
Thank you for these excellent talks. Amos Het from Perspective Therapeutics. We're developing a lead-212 actually based targeted alpha therapy for fibroblast activation protein, which was mentioned in the talks. I guess my question is for maybe for Zach and the other panelists, but really around the immunological sort of status of sarcomas, whether there's a significant difference across the sarcomas. I know it's many different diseases, right? So I'm sure there's some that are more radiosensitive than others to things like external beam, but in the RLT space, what's the range of immune type environments that we see across sarcomas?
So others in the room, I'm sure, better expert to answer that than I, but it's wide-ranging, I would say. That's probably true for almost all questions about sarcoma. It's wide-ranging. There are folks who respond to checkpoint inhibition, so there's that contingent, but in general, there's many that are translocation-driven and fairly limited in mutation burden and poorly immunogenic, and that is one area where, just to take back to an earlier question, most of our mouse models don't very well replicate truly poorly immunogenic settings. And there are many limitations to that I think the answer is not necessarily to cut off mouse testing entirely, but to leverage our companion canine species, which do often recapitulate immunogenicity of human tumors, and to do deeper dives in phase 1 testing clinically with an expectation that we may need to iterate there because many of these things I think could translate if we get it right, but there's many ways that we could miss potentially effective drugs or combination approaches if we don't take the time to do careful science at the level of phase 1 studies.
So maybe I'll just pivot back to your question, because I think one of the coolest things you showed in your models was that depending on the amount of radiation that you are actually exposing different niches of the tumor to, you're getting different immune signals, different early immune responses. So some of the things we all hope for, the damage-associated patterns, type 1 interferons, all the good stuff that we think might be able to turn a cold tumor hot. So if you were designing a study with one of these particles for patients and recognizing, again, to your point, this crazy heterogeneity, what do you combine these particles with? And in terms of immunotherapy, is PD-1 enough because you just hope that it somehow, somewhere along the way, something teaches a T cell how to get there? Or do we really need to be dissecting down a little bit more to more spatial approaches or some other type of cell therapy or something to that effect?
Yeah,
or bispecific, maybe.
I think there'll be, not one size, one response, one combination that addresses all of that for sure. And so I have designed a study, for example, and I think, one population that might be a lower hanging fruit for a radiopharmaceutical combination that our data would be consistent with is those patients who did mount a response following a checkpoint inhibitor and then developed acquired resistance. So there's evidence that they have the capacity for their tumor to be recognized, and it has been. And reigniting an existing response is, a lower bar than a patient who never responded, has de novo resistance. And some of those may be better suited to, synthetic immunity with CAR T cells or other cell therapies of that type. And these are testable questions even in the clinical setting where, with a bit of a deeper dive on tracking adaptive responses, you could have biomarkers that would identify cohorts where there is a response or is not. I think the field over the next 20 years will be in that position where you could delineate what type of immune cold patient you have at this point in time, and then you may have to tailor the combination to that.
Okay. Many thanks to Dr. Wilkie, these wonderful presentations and a very enlightening discussion.